So for our last speaker, we have Robert talking about how building genomics tools and taking command line tools and bringing it to the browser has improved performance. And I'm super excited about this talk. So give a round warm welcome for Robert.
All right. Thank you, Jonathan. So my name is Robert Aboukhalil, and I'm a bioinformatics software engineer, and I work at the genetic testing company called Invitae. Today, what I wanted to tell you about is how we can try to bridge the gap that exists between really fast command line tools and really interactive web applications. But before I tell you about this, it always helps to talk about what is WebAssembly exactly. So in a nutshell, WebAssembly is simply a binary format that encodes an abstract syntax tree instruction set intended as a compilation target for low-level languages, obviously. Now, let's do this again, but in English, it's really just a new language that is capable of running in the browser alongside JavaScript and lets you do things like take existing code written in other languages and compile them using tools like Emscripten to WebAssembly. So far, it's been used for things like gaming, porting simulations and 3D animations, and porting entire desktop applications to the web. So it's been super powerful. One thing it hasn't been used for as much is data analysis and data-heavy web applications. To understand where WebAssembly would fit into this realm, let's think about what exactly makes a web tool. So typically, there are two portions. One is the data analysis, and this is where the number crunching is going to happen. And then there's a data exploration, which is where you want to interactively explore your input and output data. Typically, the way we've mapped this is by running the data on the server and by doing the data exploration in the browser. And that's worked fine for most cases. But sometimes you really want to make your apps more interactive. And you want to avoid that extra round trip to the server, so you really want to do analysis in the browser as well. So sure, you can use JavaScript, but that comes with a set of challenges. One, it's typically going to be slower than your existing tools that are running on the server. Two, you're going to have to rewrite the tool in JavaScript to begin with, which is not fun. And third, there's the issue of how do you make sure the browser doesn't just crash because it's doing too much computation. So the way we've approached this is by trying to leverage WebAssembly in the browser to try to address some of the speed issues and the portability issues that we've seen with JavaScript. And to address the responsiveness, you can leverage tools like WebWorkers to run JavaScript code in a separate thread so that the UI stays responsive. And so this allows us to run the actual WebAssembly heavy computing in a separate thread. So let's look at a concrete example of how this would happen. This is an application that we developed about a year ago, and essentially what it allows you to do is take some DNA sequencing data and look at the quality of it. Get a first glance at did the sequencing run go well or did it not? For this to make any sense, I'm going to give you a very brief introduction to DNA sequencing. So the idea is that you have a bunch of DNA molecules, and when you want to read off the letters in the DNA, the A, C's, T's, and G's, most technologies, or actually no technology currently available, allows you to read the whole molecule. So you have to chop it up into smaller fragments. And these DNA fragments get pumped into the sequencing instrument, which outputs a fastq file. This fastq file contains the DNA sequences of all the fragments. And what this fastq.bio tool does is it takes this fastq output, does some number crunching. It tells you, here's the quality of this file. It gives you a bunch of interactive graphs. So you would go into the website, you drag and drop a fastq file. It would interactively sample through the file and calculate metrics and allow you to interact with these graphs. And the way we had written it was just in JavaScript. Although there are half a dozen command line tools that do the exact same thing, we had to rewrite it to JavaScript, which was very painful and error-prone. So in terms of the architecture, given the fastq file, we would have a main controller that would essentially sample random chunks of the file and then calculate some QC metrics on it, and then plot the results. And then it would go to the next chunk of the file, and so on. So this was a pretty simple architecture. What we wanted to do, though, is to get rid of this red box that does the calculate QC metrics. And instead of doing that in JavaScript, we wanted to use this command line utility that's very commonly used in bioinformatics called seqtk. And it stands for Sequencing Toolkit. And it helps you manipulate sequencing data, but it also gives you quality control metrics, which is nice because somebody spent the time to write it, and it's written in C, so we're going to take it and compile it to WebAssembly. And just to show you exactly what we had to do, on the left, I show you how to compile seqtk to binary. You use GCC, and you use a bunch of flags according to the README file. Instead, you're going to use Emscripten, or EMCC, and then compile it to WebAssembly. Make sure we have support for the zlib library so that we can take in gzipped fastq files. And then we're going to ask Emscripten to give us a virtual file system. This will allow us to mount files virtually within the browser in memory. And the reason why that's really useful for things like genomics is because everything happens with files. So if you look at how seqtk is called, you would call the seqtk program and you might say fastq check followed by the name of the file. Instead of doing that, we're going to do this in JavaScript using Emscripten's utility function call main, and we're going to give it the exact same parameters. So this virtual file system would allow us to take chunks of the file, mount it to that file system, and then just call the exact same command without having to rewrite the C code so that it doesn't work with files but strings instead. So now the main controller for added responsiveness is going to interact with a WebWorker controller that's in charge of creating and maintaining the state of this separate WebWorker. Now the thing with WebWorkers is you can't just say, I want to execute this function within the worker. You can only communicate with messages. So you essentially need an API that allows you to interpret the messages that you get from the main thread and do things like mount some chunk of the file to the file system, sample through the file randomly, execute a C function from the WebAssembly, and so on. And so once it does that, it returns a promise to the main thread. And once the promise resolves, it essentially plots the results again and keeps going. Because this is a relatively common pattern in genomics, that you want to sample files, calculate a QC metric, and then show it to the user, we've kind of wrapped this around in this tool that we call Aioli, which is available on NPM, one of the 0.06%, as we heard earlier. And so it takes care of abstracting out all the message passing and the setup for you. So if you find yourself, even outside of genomics, needing this kind of pattern, please check it out. It's open source. OK, great. So now we have an architecture. What does this actually look like in practice? So on the left, I'm going to show you a screencast of the JavaScript version. And on the right, I'll show you the WebAssembly version. So what we're going to do is go to the website, choose a fastq file in both, and then you can see the plots are starting to get generated. Now, if I pause it here, you'll see that the WebAssembly one has done already over 25,000 reads, whereas the JavaScript one is barely done sampling through 3,000. So clearly, it's faster. It feels more responsive. But let's actually calculate that. And the way we're going to calculate that is using the metric of how many reads per second. You can essentially think of that as how many files, how many lines per second can you parse in the file and calculate on. In our original JavaScript implementation, we could do about 1,000 reads a second. So what we did then is we took the seqtk command line utility, straight out of the box, compile it to WebAssembly, and then using Aioli, leverage WebWorkers, we're able to get about a 9x speedup without modifying the C code at all. which is pretty awesome. But then we noticed that seek.tk outputs a lot of really useful information, but that we weren't actually using some of it. So we went back to the C code, modified it, took out all the parts that we weren't using in our plots, and now we suddenly see a 13x speedup. Now, at this point, the way we've been running or generating these plots is by calling two separate functions in seek.tk. So we went into the C code again and modified those two functions and combined them essentially into one. What this means is that instead of reading the file twice, one for each function, now we just parse it once and execute the same command on it. And with that, now we're at 21x speedup compared to the original implementation. And before I go any further, just a few caveats. This 20x speedup is not generalizable to all tools. I don't want you to come out of here thinking, I'm going to use WebAssembly and get a 20x speedup. You might only get a 2x speedup. You might only get a 10% speedup. You might even get a slowdown if there's so much communication that needs to happen between the main thread and the WebAssembly that you just end up spending all your time there instead of the analysis. The other thing that you might note is that this original JavaScript version, we didn't optimize. And so this might be an argument against using it as a baseline. But to me, that's where the power of WebAssembly comes. We didn't have to bother spending time optimizing the JavaScript. Even just taking an off-the-shelf tool directly, we get a 9x speedup compared to a reasonably adequate implementation. Now, of course, the real question is, well, it's faster than our JavaScript implementation. How does it compare to the native runtime? So we've done this comparison with Firefox and Chrome, and it turns out that the command line is still about 10 times faster than running it in the browser with WebAssembly. So this tells me that there's still a lot of room for improvement where we can get a little closer to that 1x. And in particular, when you're talking about tools like this, they're very file intensive, and they operate on small chunks of file, and they have to communicate a lot. There's tons of stuff that we could do to reduce this gap. So while we've been working on WebAssembly, some of the things that we noticed that they're good for, one big category, of course, we all know is games, supporting games to the browser. So for example, here I took a Pac-Man clone that somebody wrote in C++. I used Emscripten to compile it to WebAssembly, and now you have Pac-Man running in Firefox. And it works great. It works super fast. It's a little challenging when you're dealing with games to port them, but when you do get it to work, it's pretty exciting. The other category is data tools. So I've mentioned how you can do image processing. We talked about fastq.bio, doing data analysis in the browser. Another really interesting application is if your web app requires users to upload a large set of data to the cloud. While the upload is going, what you could do is start pre-validating, maybe even looking at the QC of that data so that you could tell users, you know, you're uploading this 30 gigabyte file, but it doesn't look like the quality is that great, or it doesn't look like you're meeting the format specifications. And this is especially great if you're dealing with binary files that have a complex enough structure that there are already command line tools that are existing that let you do this sort of stuff. The last category that I think is underexplored is educational tools. So to give you an example, you can imagine that you can take command line utilities, compile them to WebAssembly, and use them essentially as a teaching tool. So just to give you An example, I recently developed JQ Kung Fu. This lets you This essentially takes JQ and compiles it to WebAssembly and has it running in your browser. So you can specify a query, an input JSON, and an output JSON. And once the WebAssembly is loaded, it is super fast. And although there are other websites like this, they're typically implemented as I'm running JQ on the server, and I have an API call that I make to essentially execute the command and then bring it back to the user. The issue there is, one is speed, because you have to go to the network. The other issue is security, because if I'm hosting this application as a backend application, I need to make sure the user can't execute any JQ query on my file system. And really, the other advantage is convenience. This app is entirely front end. It's literally just a bunch of static files hosted on Google Cloud Platform. So this list wouldn't be complete if I didn't talk about when you really shouldn't use WebAssembly. There are a few use cases that we came across that are particularly bad. One is if you're looking to analyze an entire file, and that file is really large. So let me give you an example. If you have a file that contains some matrix, and you want to calculate the pairwise distance matrix of that file, to do that, you have to look at all the data at once. And so if that file is large, that's just not going to be something that you want to do in the browser. But with fastq.bio, we only looked at chunks in the file at a time and could calculate things independently. So that's where it really shined through. Second, if we need large reference files that are loaded into memory. So in genomics, we need this a lot, where we have, let's say, the human reference genome, which can go anywhere from 4 gigs to 30 gigs. If you're going to deal with stuff like that, obviously WebAssembly right now can only support 32-bit addressing, so that wouldn't work. But even if it did, if you find yourself loading 30 gigs of data in your user's browser, you may want to rethink what you're doing. And the other issue is if you're doing a lot of communication back and forth between the JavaScript and the WebAssembly, that is definitely going to be a killer. Okay, so in short, we've talked about how using WebAssembly, out of the box, we were able to get a 9x speedup, and with some modification from the C code and optimization, we could get about 20x speedup for fastq.bio. We also talked about how using a web worker is pretty nice because you can maintain the responsiveness of your application. Finally, shameless promotion here. I recently published a book called Level Up with WebAssembly. So if you're interested in learning about how to get started with WebAssembly and the ins and outs of how to use it, check out levelupwasm.com. And with that, I want to thank you for being here.
We have time for a few questions for Robert. And we have about 15 minutes before we get shuffled out of here. So maybe quick ones.
So you talked about performance as one of the main benefits. Did you see any other benefits as well? Like, I know you were dealing with some strings. Did you have your JavaScript run out of memory? Or what kinds of other issues did you see with, you mentioned browsers crashing.
Right, so in terms of benefits, I would say for this category of problem, speed is a big one. Portability might even be a bigger one. So in a field like genomics, a lot of the tools have to be really optimal, and so they tend to be written in C or C++. So the ability to take all this existing knowledge and port it to the web is even more important in a scientific field because you don't have to spend time revalidating that I rewrote the JavaScript correctly. And it also means that the community, the scientific community, trusts that, okay, this application is using a trusted tool. In terms of, you mentioned the browser crashing. What I was referring to was that if you run all your analysis in the main thread, whether it's JavaScript or WebAssembly, you're going to end up having your browser telling you, okay, wait, I'm running out of, like, this application is taking too long, or your scroll bar is going to freeze. And so that's why the WebWorkers can help on that front.
Hi. I liked how you separated the pros and cons list of like when WASM is good and when it's not. And one of the ones that stuck out to me was you said large files. Now, I kind of was wondering if you made some assumptions there, because you can put all of your data in WASM linear memory, or you could use other tricks like typed arrays in JavaScript. And I was wondering if you have a comment about that.
Sure. So in fastq.bio, we don't read the whole file. So you can and give a file as big as you want. It will sample through the file randomly using the file reader API, so it doesn't have to read it all in a string. And the nice thing about the WebWorkers, actually, another good reason for that, is that with WebWorkers, you can mount file objects to the virtual file system. So you don't have to, whereas outside of WebWorkers, you need to load it into a string and then mount it. So using that approach, you don't actually need to read the whole file into memory. The place where it gets tricky is if you do need to look at the entirety of the file at once when you do an analysis, which luckily wasn't the case with fastq.bio.
Hi. Is there any benefit for security using WASM whatsoever?
Yes. So one of the really cool things, so again, I'm going to go to fastq.bio. So this application is available offline once you load it in your browser. This has implications for people who work in clinics who don't want to upload their data to a server and have it analyzed. They just want to see it in the browser. So from that perspective, yes. I'm not super familiar with other applications, but it sounds to me like WebAssembly is fairly restrictive as to what you can do. So, given the linear memory model, and you can't really go outside of that. So I think there's other uses there.
Yeah, one thing I've read is that the heap and the stack are actually separated. And in classic binary, that is not the case. So things like buffer overflow and running through the stack. So my interpretation of that is that you can no longer do that kind of exploits. So do you confirm that? Can you shed some light?
I would not. So I would not confirm that because I recently came across a blog post that claimed that it could circumvent some of these things, and I don't understand too much of the details myself, so I wouldn't be able to speak to it. But yeah.
One of these days. There we go. I think that's the last question we have for tonight. I want to thank Robert again for speaking. That was an awesome talk. So round of applause for Robert. Thank you.